This is a new paper I've been hoping to see for a long time. Some of the most amazing Cambrian fossil preservation extended into the Ordovician. So, does it tell us as much as we hoped? Short answer, yeah, it pretty much does. Even the most extraordinary fossil deposits have biases and limitations involved, whether that's because of the type of preservation that's going on and the process that leads to it, or just because of the environment that it's sampling. No one deposit can tell us everything. So let's take the Burgess Shale type faunas to start with as an example. These are probably the best windows that we have into ancient life, in that they preserve almost everything that was alive on the seafloor at the time of burial. There are numerous really amazing deposits now from Cambrian rocks and even extending into the Ordovician, and the best of them preserve fully soft-bodied animals as compressions of carbon on bedding planes in mudstone, sometimes with really extraordinary detail. These faunas have revealed a surprisingly consistent ecosystem around the world. In many cases, the same major groups are present, in much the same proportions, and surprisingly often we even get the same genera shared across continents around the world, through relatively long periods of time, through the early and middle Cambrian. They've revealed what looks like a consistent pattern of how these ecosystems were structured. And they've made us actually think that we kind of understand Cambrian communities, at least to this extent in the offshore mudstone facies. So, where's the bias here? It comes down to the environment that they're being preserved in. Because as far as we can make out, and this is mainly work by Bob Gaines, who's done a lot of really good research on this, these deposits rely on the animals being washed from an aerobic environment where they were living to a slightly deeper and anoxic environment. So although it's not just anoxia that causes the preservation, that seems to be a major factor. And you can only get anoxia on the seafloor, in general, where the water is still. So it has to be below the depth that waves will reach and churn it up and bring oxygen down to the seafloor. So that puts all of these faunas more or less at storm wave base, which is the deepest level that storms can reach, and it's normally between 50 and 100 metres deep, depending on the geography. Pretty much all of the Burgess Shale type faunas record that sort of environment. If there were other things that were living further up the shelf in the much more turbulent, agitated waters and were not being transported out into the deep, then they would decay. They would be destroyed by being washed around in these conditions, and we wouldn't know they were there. That's where Nick Butterfield comes in. He pioneered a new technique and discovered a parallel set of exceptional preservation in Cambrian mudstones, which he called small carbonaceous fossils, or SCFs. These are mostly recovered from aerobic, shallow water deposits, where the Burgess Shale type faunas can't actually reach. And so, as a result, they're quite different. For one thing, they don't preserve whole organisms. And they don't preserve truly soft tissues either. What they do preserve, though, is extraordinarily detailed fragments of unbiomineralized, so soft-bodied but not completely squidgy animals, or just the elements of them that could fossilize, so the radula, the sort of scraping mouthparts of mollusks, for example, or little cuticular plates from arthropods. So these remains have to be toughened to some degree, sclerotized, we call it. But because of that, when the animals decay or are eaten or washed around, you do get these bits left over, and they don't tend to be completely destroyed. And it's these type of remains which are pretty much as they were when they left the animal, extraordinarily detailed and exquisitely preserved. It's these types of remains that are found by this technique. So the SCFs are extracted by dissolving mudstones in hydrofluoric acid, which is something that Lucy won't let me do in the kitchen, unfortunately. But such is life, we get by. But you have to do this in an extraordinarily careful and delicate way, with no agitation, otherwise the things get broken up. Watnick and then his student Tom Harvey, who has gone on to a career in paleontology as well, and now other groups around the world have been managing to achieve, is that when you use this protocol, you can discover previously unanticipated pieces of incredible detail from long extinct animals. You don't find SCFs everywhere, but where they were preserved, they reveal a whole new window on Cambrian life. While some of the most common elements of them, things like Wiwaxiid sclerites and bits of the parapulid worm or toya, tie in very neatly with things that are also common in the Burgess Shale type faunas, there are also other things alongside them. And this is where we get pieces of things like copepod and branchiopod crustaceans, which were not known to exist at this time, and they've been preserved as their diagnostic mouthparts in these SCFs. So the SCFs are showing us that alongside all of these relatively early stem group type arthropods that we have in the Burgess Shale type faunas, we also had much more modern groups evolving in parallel alongside things like loriciferan worms, which again have virtually no other fossil record. These things were turning up and evolving and diversifying in the shallower water. That's an amazing thing. But I've been waiting for people to extend this technique into the Ordovician. During the Ordovician, we have a relatively limited array of exceptional preservation. We don't have many Burgess Shale type deposits, and most of them are limited in one way or another. Most of the other exceptional deposits that we have record either very specific marginal marine ecosystems or very deep water communities. We know that there was a spectacular diversification of life going on and radical changes in paleoecology, but most of that is based on things like brachiopods, trilobites, corals, all these things with biomineralized skeletons that leave a good fossil record. So SCFs potentially offer us another way of assessing what was going on in life during the Ordovician. We even had some colleagues in Sweden, Elise Vallet and Seb Willman, doing some work on this for the Castle Bank biota, which was published in that first announcement paper. And they did manage to recover some SCFs, but they weren't spectacularly good, and yeah, they weren't as good as the Cambrian ones, shall we say. So we've been starting to wonder, was there a window in the Cambrian for exceptional SCF preservation, as there was mostly for the Burgess Shale preservation? And did that window close before we got to the Ordovician? Well, finally we know. It was still open. Nick's current PhD student, Giovanni Massini, has been producing some really excellent work over the past few years, and he leads on this paper. So it describes two suites of fossils from boreholes taken in North Dakota. They have called these the Osterberg biotas, and they were deposited in very warm, shallow seas, what we call epeiric seas, basically flooded continental crust, and these are the hotspots of biodiversity in the modern world, and probably for most of the past as well. There are multiple samples from each site, a total of 38 samples, and only one of them failed to produce SCFs of some sort, which is really good going. It means these are widespread fossils through these formations. They appear to have sampled sandstones, siltstones and mudstones, although it's not entirely clear what level was which, what the lithology was at each thin bed, but it looks like all of these different rock types have been producing some quite nice material. The sediment is also often burrowed, apparently, showing that this was highly aerobic. There were lots of things churning through the sediment, eating any remains, and some of these remains may even have passed through animals and out the other end. So they're quite tough bits of organic material, and they are exquisitely well preserved. The first of these two intervals covers the latest Cambrian into the early Ordovician, so it's the same age as the rocks leading up to the Fezwata biota in Morocco. Then the other interval is after a big unconformity and is in the middle Ordovician and very close to the age of Castle Bank biota. So this is really handy. In terms of the level of detail in these fossils, what they've uncovered is on a par with the best of the Cambrian SCF faunas. and that's saying something. But these are really exquisite. So, for example, there are a whole array of molluscan radulae. These are the scraping organs that are used by mollusks and all their relatives of mollusk kind, including ones from predators, which are probably assignable to nautiloids. That shouldn't be a surprise, since nautiloids were really getting going during the Ordovician. But they have enough features in them, in the little hooked teeth, to be able to recognise that these are ones from predators. Then they also have the jaws and other fragments of crustaceans, including possibly the earliest eumalacostracans. This is the major group that includes most of the familiar crustaceans today, the crabs and lobsters and so on. But those would be the earliest known. They also have fragments of the breathing apparatus of eurypterids, sea scorpions, and these go back even into the older of their samples, showing that these also are the oldest known examples of eurypterids. There are lots of cuticle fragments from scalidopteran worms. These are priapulids and their relatives, which produce distinctive spine-like structures which are embedded in their cuticles, and including in their proboscis, which they use for feeding. And some of these are from groups that are microphagous. They feed on extremely tiny particles of food rather than being predators going after things that are barely smaller than they are. It's a wonderful suite of fossils. This shows us that this type of preservation, the window stayed open until well into the Ordovician, and probably therefore much, much later. This offers us opportunities to track this sort of preservation through most of the Phanerozoic, potentially. It's just that nobody has really looked yet, as far as I know. But the main message of this paper is about the ecology. And this figure here is the most important in terms of understanding that message. There's a lot of information in it, but there's a few things particularly want to note. Firstly, it's divided into three major sections. So the top part are the Burgess Shale type faunas, i.e. animals that are preserved entire but also recognisable as fragments in SCFs. Then you've got the shelf fauna, which is found exclusively in SCFs, and then an Ordovician type fauna of animals that didn't exist in the Cambrian. So when you start comparing these, you get some very interesting patterns. The shelf fauna is largely continuous, well into the Ordovician. The same groups are turning up from the Cambrian deposits and the Osterberg faunas. This is really interesting. It suggests that there's quite a lot of continuity here. And you look at the detail of it, there's more diversity in things like the arthropod fragments in the Ordovician ones, but the same major groups are going through. The other thing to notice is that the Burgess Shale type elements are commonly found in SCFs from the Cambrian, but hardly at all in the Ordovician, despite the same groups still being found in Burgess Shale type faunas that we do have. So that leads to the key message. The Burgess Shale type faunas were still around, but they weren't in the shallower mid-shelf environments where the SCFs are recording relatively modern groups like these various crustaceans and things like the scalidopterans that are feeding on microscopic particles. Instead, these more primitive Burgess Shale type groups, the stem groups of the arthropods and so on, now seem to be restricted to these slightly less favourable areas of the eel estate. So the slightly deeper water, or the marginal marine facies, or the South Pole in the case of the freshwater biota. And so perhaps they were being pushed out to those areas by the evolution of these modern groups in the areas that are being sampled by the SCF communities. The principle of this is not a new idea. It actually goes back to the 1970s early work of Simon Conway Morris, and has been reanimated a bit more recently with the Llanfairllteg biota, where you have Burgess Shale type survivors in quite deep water in the early middle Ordovician. However, what we've previously been thinking of is that it's a much more extreme thing, that you get these refugia in very deep water or very cold conditions, rather than it being a much more subtle compartmentalization of the shelf into the most desirable bits, which are the warmest and turbulent and aerobic, and the ones which are just pushed off slightly to the side where the oxygen levels get a little bit lower, and so on. and therefore this affects the areas around storm wave base. And this is, as far as I can make out, it's quite a new idea, and this is something that is going to affect our thinking when it comes to things like Castlebank. I have got a couple of caveats about the whole thing though, of course. One of them is that we do have to remember that no matter how wonderful these fragments are in the SCFs, like all faunas, these are going to be biased. And in this case, we're not seeing the wide range of soft-bodied animals that don't have any sclerotized bits that could fossilize in this way. So we have to always remember that while this is telling us about the components of the fauna that can produce SCFs, it doesn't tell us anything about what might have been happening to the groups that didn't. And that could be a very large part of the story, in the same way that the soft-bodied faunas are a huge part of the total ecology, when normally we can only see the skeletonized taxa. The other potential fly in the ointment here is that Castlebank seems to show a bit of a mixed fauna. Rather than being just Burgess Shale type elements, there are also numerous small crustaceans or crustacean-like arthropods, and various tiny worms such as chinorhynchs, and some of these were in the 2023 paper, but they haven't been included in the summary figures in this paper. That may well be because all we've published so far are a few preliminary pictures rather than detailed justification, and that is fair enough. But the point is that it appears that it might be a bit more complex. Rather than having the more modern groups which evolved in the more aerobic parts of the shelf, segregated from the older faunas, the ones that are surviving from the Cambrian, what we have here is a mixture that even in the sort of outlying areas around storm wave base, we've got lots of these modern groups encroaching into this community and living quite happily alongside these Cambrian holdovers. Which is interesting. You don't know whether there is an underlying dynamic there which is steadily pushing the holdovers further and further out, or whether they had created their own ecosystem in which they all worked perfectly well together, and it was other, perhaps external changes that led to eventually the loss of these Cambrian lineages at some point further down the line. Perhaps also it's a peculiarity of the Castlebank site, which, because of the small size of the animals there, is certainly a bit anomalous at first glance. Or perhaps it's part of a larger pattern that we just haven't fully revealed yet, but more work on the SCFs in future might well help that. So I just want to end this with a couple of notes of appreciation about just how difficult and time-consuming this work is. It's extremely delicate work picking out these things, then illustrating them with focus stacking, getting the contrast all right. It's a skilled job. And the other thing that often, I think, is forgotten is just the sheer amount of knowledge you need to have of modern and fossil anatomy, biology of these animals, in order to be able to recognise little bits of fragments of mandibles of different groups of crustaceans. That is not an easy job. You can't just go to your standard textbook and find a nice neat diagram showing, well, this is a copepod mandible and this is a... It doesn't work like that. You have to really know the literature in detail. And that's one of the most amazing things about the people who work on this field. Encyclopedic knowledge and the ability to apply it to these fossils. It's wonderful. And I am so glad to see this paper finally, well, say finally published. I'm sure they'll be working on it for a few years, but I've been waiting for so long for this work to be extended into the Ordovician and hopefully now to younger rocks as well, to see just how these patterns develop through the Paleozoic. And it's the dawn of an exciting new era, really, because we're dying to see what it turns up next. So, Nick, Giovanni, all the rest of you, carry on going, and good job.